So this chapter of Langman is about the development of limbs. But the first thing that you need to know is that this development starts in the 4th week, right? At approximately around the 28th day. And the development of upper limb is a little bit earlier, and the lower limb develops 1 to 2 days later than the upper limb. And the sequence in which these limbs develop is the dermatomal order, like C3, C4, C5, and so on, right? And in the lower limbs, it's from the L2, L3, L4, and so on. So in the 4th week, the first event that occurs is an outpocketing appears in the place where the future limbs would be. In the lateral wall of the embryo, an outpocketing develops. Then in the 5th week, this outpocketing, it takes the appearance of a bud. And in the upper limb, this bud is paddle shaped, and in the lower limb, it is a flipper shaped bud, right? And the next event is that chondrification centers develop and motor axons come there, and the plexuses, the brachial and the lumbosacral plexus, they develop, and the peripheral nerves appear. So basically, the skeleton is forming, and the nerves, both the sensory and the motor supply, they are entering this bud, right? Now the bud is made up of like 4 or 5 important things. The first is the core. The core is mesenchyme, right? And this mesenchyme is going to be the future limb, the bone, the muscle, the cartilage, everything is going to be formed out of this mesenchymal core, and it is derived from the somatic lateral plate mesoderm. Right, so we have different types of mesoderm. In that, the somatic lateral plate is responsible for the development of limbs. Now this mesenchymal core will have a covering, and that would be of ectoderm, and these would be cuboidal cells. The next thing is that these cuboidal cells, the most distant part of them, like the part which would be away from the body, that part is known as the apical ectodermal ridge, or simply AER, or simply the fastest growing part of the limb, right? And it is very important for bud elongation, because as this part grows, these cells multiply in these apical ectodermal ridge, and in that way the limb elongates. Now we have the core, like the center of the limb, and then we have the AER. Now in between these two parts there is a region which is known as the differentiation front, and that region separates the rapidly dividing part from the not so rapidly dividing part, right? The motor axons, they come from the spinal cord, and the sensory axons, they follow those motor axons as a guide, and neural crust cells also come, and neural crust cells, they would then turn into Schwann cells, and they would do the insulation of these nerves. Now as the bud starts growing, the bud, it came in the 5th week, and as it grows, so AER would grow very crazy, and rest of the parts would follow it. So AER is a rapidly proliferating undifferentiated zone, and the regions which are proximal to it, right, they start differentiation. Now the limb bud, it is elongating, but that elongation is undifferentiated, it's just a mass of cells, right? So the elongation is distally, but the differentiation starts proximally. So the limb would develop proximo-distally. So the parts which are closer to the axial skeleton, which are closer to the center, they would start developing first. The order is like stylopod, zygopod, and autopod. Now what is a stylopod? Stylopod is the part of the limb which would in the future become humerus, or in the lower limb, the femur. Then we have the zygopod. Zygopod is the second part of the limb, which in the upper limb would be ulna and radius, and in the lower limb it would be the tibia and the fibula. And the autopod is the last part to differentiate, and this is the future hand, or the carpals and metacarpals and phalanges in the upper limb, and the tarsals and metatarsals and phalanges in the lower limb. So stylopod, zygopod, and autopod. Now there are junctions between them. The junction between the stylopod and the zygopod. So what is stylopod? Arm. And what is zygopod? The forearm. So between arm and forearm, that constriction would be elbow. And between zygopod and autopod, zygopod is again forearm and autopod is the future hand. So in between them, the junction would be, or the constriction would be, the future wrist. Now this was up till week 5 that we have the mesenchymal core and we have the AER, which is rapidly growing, and differentiation has started proximo-distally. Now we come to week number 6. Here the chondrification, which had started in week 5, core for the bones, and now that hyaline cartilaginous structure is ready, right, for bone to form on it. And hand and foot plates have developed, right? Now in week 7, that cartilaginous core would start getting ossified and osteogenesis begins. And primary ossification centers start appearing there, and the rotation of the limbs also happens in the 7th week. Then in week number 8, the fingers and the toes would appear after the process of apoptosis, and finally the limb would start looking like a proper limb. And by week 12, all the bones would have their ossification centers. So in summary, what happened was that in week 4 there was an outpocket. In week 5 it became a bud. That bud had chondrification. In week number 6, that cartilaginous structure completed and the hand and foot plates were formed. Then in week 7, on the cartilage, calcification happened and the ossification centers, they appeared and the limbs were rotated. And in week 8, fingers and toes were formed, and in week 12, all the primary ossification centers were complete. Now there are some genes that you need to know about the limbs. The first one is the FGF10. That is the gene which is responsible for the bud formation. Then we have TBX5 and TBX4. So 5 is for the upper limb and 4 is for the lower limb. Then we have the gene homeobox or Hox gene. So I told you development occurs proximo-distally or craniocaudally, you can say. So that craniocaudal axis is established by homeobox. Then we have FGF4 and FGF8. They maintain that progress zone that is our AER. And the retinoic acid is basically an antagonist to that progress zone, right? So these two genes, they work in harmony to bring about an approximate level of differentiation or an approximate level of progress, neither too much nor too less. Then the differentiation front is the border between these two, and at the between the AER and the rest of the limb there was differentiation front, right? So it's that. Then we have MEIS1 gene, HOXA11 and HOXA13. MEIS1 gene is for the stylopod which forms the first part, arm, or HOXA11 is the zygopod means forearm, and HOXA13 is for the hand, autopod, right? Now the clinicals, so in the clinicals remember two things, milia and dactyly, right? So milia means limb and dactyly means the digits, your fingers, and a limb is like the upper limb, lower limb, so like that. So meromelia is an absent limb, but not full limb is absent, so it's absent but partially absent, right? Amelia is when the full limb is absent. Then there is phocomelia when the bones in the limb are absent, and it's also a type of meromelia. Then we have micromelia where the limbs are present but they are short. Now we have dactyles, so dactyles means digit, brachydactyly short digits, and arachnodactyly is long digits. Then we have syndactyly, syn means fusion, so some digits they get fused, that's syndactyly, and then we have polydactyly and ectrodactyly. So polydactyly is an extra digit, like instead of 5 fingers they have 6 fingers, and ectrodactyly is a less digit, instead of 5 they have 4 fingers. Cleft foot or cleft hand, the third digits are missing, the middle finger is missing, right? Next is a TBX5 mutation. So TBX5, you know, it is for the upper limb, and in this mutation, as a result of this mutation, the upper limb would have defects, but along with that, heart defects will also be present, and this mutation is termed as the Holt-Oram syndrome. Now next we have the AER anomaly. AER, which was our rapidly dividing zone. So when there's an anomaly in that zone, there is a deficiency, transverse limb deficiency. So the lower part of the limb is missing, right? because that front was responsible for the elongation of limb, right? So the limb would differentiate, but it would not elongate. And that is why the lower part of the limb would be missing. Next is that amniotic band, sometime in the womb, during development of the child, these the amniotic membrane, it has some loose bands, and they wrap around the child in a way that they wrap around its digits and limbs, and because of that the digits and limbs cannot develop properly and they cannot grow properly and they remain constricted. So when the child is born, either his digits are deformed or the limb is missing and things like that. So those are because of the amniotic bands. And then we have PIT-X1 mutation. So PIT-X1 gene, along with the TBX4 gene, it is responsible for the lower limb formation. So because of this mutation, there would be clubfoot. Fibrillin gene mutation, we all know that's Marfan syndrome. The person would have long digits, right? Erectrodactyly, right? And when there is a type 1 collagen mutation, so there is osteogenesis imperfecta, the condition, and because of that, the bones would be very fragile and they would be very susceptible to frequent fractures and hypomineralization. That's all for the development of limbs.